Well, welcome everybody. I'm Patrick Brown, a professor of plant sciences at the University of California, Davis. And I had the pleasure of talking to you today about boron in plants and agriculture. Now, my goal for today is to give you enough of an understanding of how boron functions in plants, in soil, in agriculture, so that you, whether you're a grower or a consultant or a student of agriculture, can understand how we make the decisions that we make. Now, as I go through this, there will be 5 separate sections, each of which has a potential role in helping you understand how boron functions and how we make the decisions that we have to make in the field. I'm going to start with a history and a little bit of an overview of the occurrence of boron deficiency in soils and crops. And I think there you'll see how we've progressed and what we've learned over the last 100 years or so. Secondly, I'm going to take you through a number of the different symptoms of boron deficiency in different crops. And all of this is designed to give you some clues as to what boron's function is and how that leads us to the management decisions that we need to make. I will then go into some of the biology, but hopefully I can make it as clear and concise as possible. But it is important to understand what we know about the function of boron, what we know about the mechanisms of uptake of boron, and how it moves around the plant. What is the mobility of boron in plants? All of that will be tied together in the last section, where we'll talk about management strategies and approaches to managing boron, and then I'll summarize it. I will point out that I don't make specific recommendations for specific crops, and that's on purpose. This particular video will go out to a number of different countries, a number of different crops, and it would be impossible to get so specific. You'll also see as we go through this why that is actually not possible in most cases. So let's get started. Boron was discovered almost exactly 100 years ago as an essential element for plants. And the image you have there up on the right-hand side of Catherine Warrington, she was responsible for that early discovery. And she didn't discover this because she was looking for the next essential nutrient element. She discovered it because she was experimenting with changing nutrients in plants and how they affect insect attack. I'm going to show you a few of her early results and use that as an illustration of how we've progressed in our understanding over the last 100 years. Now, in my career, I've focused very heavily on boron. I've been doing this for like 35 years. And what I'm going to try and do is put the pieces together to help you understand this element so that you can make smart decisions. So let's look first at some of Catherine Warrington's early discoveries. This dates back to 1923, and she has some photographs, but of course they're in black and white because this was right at the beginning of photography. But there are a few observations she made that are really still relevant, still important. So we'll review these observations that she made, the key ones, and then as we go through, we'll see what's been added, and then we'll return to this at the end. So the first and most important point that she made was that deficiency of boron is always exhibited first in meristems, whether that's an apical meristem at the top of the shoot, reproductive meristems, flowers and fruits, or root meristems. She also made the observation, and we'll question this one a little bit later on, that boron may have a special importance in the formation of the vascular system. And her last observation was particularly important, and that is that continuous supply of boron through the lifetime of the plant is critical. I'm going to illustrate that on the image on the right. So this is a shoot of a pea plant that has undergone boron deficiency, and you can see the apical meristem has died. You can also see that some of the younger leaves have become deformed. She then transferred that plant to a plus boron solution, and you can notice the plant immediately responds and is growing very well. We're going to look at the function in that meristem. We're going to look at the function in flowering and try to explain what she exhibited here and what she has shown us. Another important contribution she made was the observation that boron deficiency has a big impact on root growth. And you can see that top image of the very short roots compared with the bottom image. All right, so now let's move on and think about where and when boron deficiency occurs in soils of the world. Now, many of you will have seen a diagram like this. This is a diagram that explains how the availability of nutrients changes with the pH of the soil. So you have pH along the bottom of that figure, and you have relative availability shown by the width of those bars. And I call your attention to boron, which is arrowed here. And you can see at low pH that bar becomes very narrow. At low pH, boron is not held by soil particles very well. That makes it subject to loss from the soil. At high pHs, boron can form some complexes in the soil, and this can also result in a deficiency of boron. But there's an occurrence that occurs at high pHs that I'll also speak to a little bit. One of the most important determinants of whether boron in a particular environment will be deficient or not is irrigation and rainfall. And irrigation that contains boron and rainfall that has very low levels of boron is often the most critical factor to determine whether you will have a deficiency or a toxicity. Leaching occurs in the humid, wet environments. Boron can accumulate in dry environments. To summarize some of these points, first, boron is the second most prevalent micronutrient deficiency around the world. Deficiency is most likely when you have low pH, acidic soils, particularly if they're light textured, because boron can be leached from those soils. While boron can be present in organic matter, boron in organic matter is very complicated, and it's very source dependent, very age dependent. At the other end of the pH scale, as boron in high pH circumstances, boron can be tightly bound to organic matter. It can be tightly bound to a number of soil minerals, calcite, clay, laterite. And as I mentioned before, one of the most important considerations when managing or thinking about boron in agricultural environments is water, whether it's rainfall or it's irrigation. Water is often the primary determinant of boron status. In conditions of high rainfall or high irrigation with a low B water, in well-drained soils, you can easily get leaching of boron from the soil, resulting in a boron deficiency. The opposite of that is irrigation with water high in boron, particularly in arid zones, where you can get an accumulation of boron, resulting in toxicity. Now, there have been a number of efforts to make maps of boron deficiency around the world, and I show a few of them here. Starting in the top right is an old map from the 60s by Victor Shorrocks at the Borax Corporation, showing very crudely regions of high potential for boron deficiency. Below that is a map that I got from colleagues in Canada that is the result of mapping 20 to 40,000 individual soil samples in Saskatchewan, Alberta, and Manitoba. And you can see the variability in boron that occurs across those landscapes. Below that is a map from David Franzen at North Dakota State University, showing how diverse and variable boron is, even over quite small areas. All of that illustrates how difficult it is to get a good soil map, and one that can really tell us what to expect. The large map that I show is simply a map of soil acidity. And there is one circumstance where soil conditions can be very predictive of a boron deficiency. So boron deficiency is most prevalent, as I said previously, in the low pH soils, particularly those soils with high rainfall, such as Brazil, such as regions of the northeast part of the US and northeast portions of Asia. But that's not the whole story, and we'll get into some of those details in a moment. But just to summarize, there are relatively few good maps of boron in soils. And the reason they're ineffective is because there are so many other factors that are critical in determining whether a particular soil, a particular location, will suffer from boron deficiency. Crop types, the environment you're growing in, irrigation interactions, all of those act to prevent us from making very good generic recommendations. Finally, there's also a challenge that we have with how we do our soil tests and our plant tests and how we interpret them. I'm going to illustrate a couple of those points in the next slides. Many of you, of course, are familiar with soil tests, the process whereby we collect a soil from a field, expose it to some chemical treatments, derive a boron concentration or nutrient concentration, and use that information to make a decision. Now, there are some fundamental rules that are true for all elements, and the first one is that all nutrients that plants need are taken up from the soil solution. What a soil test is designed to do is to measure how much of that nutrient is in the soil solution and how rapidly that soil solution will be replaced by nutrients stored in the solid phase, the labile phase, associated directly with soil particles. This is the foundation for all soil tests. Now, over the years, a number of tests have been developed for boron. I show a few of them here: calcium chloride, hot water, nitric acid, etc. The question is, do they work? Do they tell us what the plant will be getting from that soil? This is an analysis conducted by some colleagues at Wageningen University in the Netherlands, who did a number of tests to see whether boron analysis from soil samples relates to plant tissue uptake. The first observation they made, and the figure on the right, is depending on the soil you're dealing with, between 70 and 90% of all the boron that is in the environment is in the soil solution phase. And this is quite unique for any element, that very heavy dependence on the soil solution phase. Very little boron is fixed. This helps explain why irrigation and rainfall has such a profound effect. Irrigation and rainfall dilute and leach boron through that solution phase. They subsequently did a number of tests looking at how the measured boron in the soil test correlates with plant yield, which is of course what we want to happen. And you can see from those scatter plots whether we're talking about hot water extraction, which is on the left, nitric acid extraction in the middle, or calcium chloride. None of them show a good relationship between the measured boron in the soil test and the plant response. So this is very disappointing, but the measure, the take-home message is that boron status of soils is a difficult process. We'll come back to that, but it is something to think about as agronomists or growers or students of plant biology think about how do we make decisions in the field. There is another attribute of boron that is somewhat unique and also contributes to making our decisions more difficult. Now, this is a large study from India that I'm just using as an example, where they applied boron fertilizer to low boron soils that were growing a variety of crops. You have left the increase in yield as a consequence of the boron application, and then a number of species along the base. And you can see how variable they are. And I've circled a couple of them to highlight this. So contrasting chickpea with wheat or rice, you see chickpea is highly sensitive to boron deficiency. Sunflower is highly sensitive to boron deficiency. Onion is highly sensitive to boron deficiency. And all of these crops growing in the same soil with the same boron application. I've outlined the nature of the effect. And one of the characteristics of boron in many agricultural circumstances is that it affects flowering and seed set and reproduction more critically than it affects vegetative growth. On the far right, I've circled onion. There is a second effect of boron that's very important, and that is on storage quality, product quality. The complexity of boron also goes a little bit further. The previous slide, I showed you how crops differ in their sensitivity. But even within a species, in this case canola, different cultivars respond very differently. Now, boron deficiency is a very major problem in China. Canola is a large crop in China. And for many, many years, it was found to be very difficult to grow canola in China because the cultivars they had were hypersensitive. Subsequently, through selection and breeding, they've developed some efficient cultivars, which are shown on the right and on the left of this image, that are much better at growing in a low boron soil than the sensitive cultivar. Remember, in this particular image, all of the soil will have the same boron test, the same boron result, and yet the plants are growing very, very differently. This complicates how we think about boron, and that's a point that always has to be borne in mind. I'm going to wrap up this section and then move on to the next topic. But just to wrap up, firstly, the important take-home message is that predicting boron deficiency and crop response from soil tests is difficult. And it's difficult because of these complex interactions with the soil. It's difficult because of the interactions with rainfall and irrigation. It's difficult because cultivars and species differ. And it is also difficult because the soil tests we have and the plant tests we have are not very good at predicting if and when we'll get a response. And that is, of course, because of the 4 reasons I've shown on the screen. Climate also interacts, and we'll show you this a little bit later on as well. Now, I mentioned previously that I would not be making recommendations and guidelines, and I've got one exception here. I will put sort of generic guidelines that people use for extractable soil boron and how to interpret it. And I do this for a very important point. An example of which this might be relevant for a sensitive crop like canola. If the soil tests between 0 and 0.5 ppm, you're very likely to get a deficiency. You're probably going to get a response. And I use that word probably on purpose. You won't always. As the soil tests increase, you have more boron in the soil, deficiency is possible, but it's often characterized as an occasional, very significant response. So it's not unusual with a boron test like this to 2 years out of 4, 3 years out of 5, get a major response, but the remainder of the time having no response. That's because of some of these climatic interactions. Once you're above 1, moving on to 1.5, deficiency is unlikely, but responses are still possible. And it is only when you get to the large boron concentrations where you get into the realm where you could safely say, I will not get a response to boron. So I'm going to finish up this section here, and we will come back in the next section and discuss the symptoms of boron deficiency. And I'm going to use that to illustrate some of the principles of the biology and the management of boron. What I want to do in this section is talk about the symptoms of boron deficiency in a number of different crops. Some of them agricultural, some of them forestry. And the reason I've selected the crops that we'll be looking at today is because of the insights they will tell us into discerning what is the function of boron in plants. It'll also highlight some aspects of boron uptake and some of the environmental interactions that always have to be kept in mind when thinking about the challenges of managing boron. Start with this old diagram from Warrington in 1923, where she illustrated that apical meristem death was one of the first and most important effects. But she also pointed out that leaf shape can become deformed. If we look at a range of different cultivars, and we have here sugar beet, sunflower, legumes, and cereals, focusing first on the sunflower, sugar beet, and cereals, one of the effects you see in vegetative boron deficiency in most crops is this deformation of the leaf, as if the leaf itself cannot properly expand. And it's particularly prevalent, as seen in sugar beet and in this case sugar cane, in the apical meristem, the top of the plant. However, what is not really well illustrated in those diagrams is how sensitive the apical meristem, the growing point of the plant, is in many, many species. Now, this is an example of Pinus radiata in New Zealand, but similar plantations, of course, occur in Brazil and Finland, Canada, and the eastern seaboard of the US. And these soils where these trees are grown are typically acidic, very sandy, and very poor quality, which is why they're used for these crops. The apical meristem, which you might think is a relatively small effect. The remainder of the plant you see is quite healthy. Meristems are highly sensitive, and this very small effect can have a very profound effect on productivity. I should also point out, in these environments, this meristem death as a result of boron deficiency is much more prevalent when you have a drought or a heat spell. Now, why this is particularly important in these crops is because the death of this meristem converts a forest plantation from the high value of construction timber that relies on long, straight stems to use of this material as pulp because of the high levels of branching. Not only is the value of those two products greatly different, the cost of managing and harvesting a product that can only be used for pulp is much more expensive. Interestingly, this problem in forest crops can be solved with a single application soon after planting to prevent that death of the apical meristem and ensure you get long, straight timber. As I mentioned previously, roots are also very susceptible to boron deficiency. This is the original picture from Dr. Warrington. Here is another picture from a good colleague of mine, Ismail Çakmak from Turkey. And I put this up to illustrate a couple of important points. And what we have here are three different species, canola, soybean, and maize, grown in either the absence of boron or the presence of boron. And the first thing you can see is that species differ in their sensitivity very markedly. Canola is very sensitive, and you can see those boron deficient plants are short, very little root growth, and they have a purple color which reflects a secondary deficiency of phosphorus. Moving across to soybean, you can see a moderate effect, certainly a large effect in the roots, a smaller effect in the shoots. One thing to think about here is, of course, in most field circumstances, you would not be seeing the roots. And you'll be looking at this soybean crop and only remarking that it seems to be a little less vigorous. It'd be very difficult for a grower to detect that this is a boron deficiency. And in maize, you almost see no effects at all. There are slight effects on root growth there. What this illustrates is the great difference in sensitivity of different species to boron deficiency. It also illustrates that roots are generally the most sensitive tissue. And as a consequence, we very often don't see the first signs of boron deficiency. They're reflected only in the poor vigor and the poor success of the crop. In some species, you don't see the leaf deformation that I talked about, but rather you see a yellowing or a chlorosis. And many of you have seen symptoms like this, in this case, in metacargo and trifolium, legumes, or in tobacco, would interpret it as a deficiency of nitrogen or manganese or iron. What it really is a compromise of the root growth, which causes the plant to not be very good at getting other nutrients from the soil. So in some species, because of the prevalence of the root effect, the symptom of boron deficiency is a secondary nutrient deficiency. What also happens as those roots become deficient is nitrogen fixation cannot properly occur, and the roots themselves become leaky, which can result in increased disease susceptibility. There's one other attribute of boron which is relevant for very low pH acidic soils, and that is that boron interacts with aluminum and can make plant roots more tolerant of an aluminum toxicity. 3 images on the right on this screen, they're all exposed to the same amount of aluminum, but you increase the amount of boron as you go from A to B to C. The red area you see are dead cells, and you can see clearly that as the boron is increased, the amount of damage caused by aluminum is decreased. I'm going to move now to what is the most important effect of boron deficiency, and that is on reproduction. And of course, for many, many crops we grow, This is what we are trying to do. We're trying to grow seeds or fruit, grain, etc. I show a picture here of pistachio, which I know many of you would not be familiar with, but you'll notice what's happened here is that the flowering rachis, the flowering structure, has died. And it's died even though the leaves are still quite healthy. This is a principle we'll look at in a moment. Here's a number of species, all showing effects of boron deficiency. Canola at the top, where boron deficiency leads to very poor seed set, pod shattering. The second picture below that is avocado, but a similar effect is seen in coffee, mango, pistachio, and a number of fruit and nut crops, and oil palm, where the flower itself, the female flower, is very poorly formed and fruit set doesn't occur. And on the right is maize. And in maize, boron deficiency affects both the male and the female. Boron deficiency is known to affect pollination, cause sterility of flowers, and cause abortion of both the flower and the fruit set. One of the important messages from this is that in many cases, the symptoms of boron deficiency are occurring in tiny organs, like the flowers, like the pollen tube, and only for a very short window of time, the effects of which are very profound for the lifetime of the crop. I'm going to focus on a couple of examples of this, and I'm focusing on examples and species that will share with you some important messages. This is flower pistachio. One of the important considerations is pistachio flowers in the springtime, and we see commonly when the winter has been particularly cold, the soils are cold, and then we get a warm spring, causing buds to burst and growth to occur very rapidly. That represents a stress. Boron can cause damage. In this case, you can see it clearly, death of the flowers, while the leaves are perfectly healthy. The boron requirement is clearly this very narrow window, but in a crop like pistachio or any of the fruit crops, not setting your fruit does, of course, have a profound effect for the remainder of the cropping system. The yield effects can therefore be quite dramatic. I'm going to show you one more example of how sensitive species can be to boron deficiency. These are tobacco plants. This is a lab experiment in which we grew the tobacco in the presence of adequate boron and then moved them to a solution with no boron. And within 12 hours of that transition, all of the flowers subside and fall off, and the tobacco plant sets no seed. So a very rapid effect, a very important effect. An effect of boron deficiency on flowering and seed set in a matter of hours. Interestingly, even though there is boron in those old leaves that you can see, that boron is not available to help the plant get through this deficiency. Short-term deficiencies can have really profound effects that can't be satisfied by reserves of boron that exist in the plant. Sunflower, I put this one up to illustrate interaction between the environment and boron deficiencies. The plant on the left, you can see symptoms of boron deficiency in those crinkled leaves at the base of the plant. Then you see the plant is growing perfectly well beyond that. This reflects a period in the growth of this plant in which boron deficiency occurred. And in this particular case, this is because it went through a drought. If you look at the sunflower on the top right and the one below it, we can see also that imposition of a boron deficiency, again through drought, results in the individual flowers of the sunflower head not setting seed. It tells you that variable soil boron supply can result in very intermittent symptoms, and depending when they occur, this can be super important. Also, it shows you how these things can be exacerbated by drought and heat, which compromise water uptake. One additional example is in wheat, and I'm drawing examples from all over the world. This is from Bangladesh. And in Bangladesh, they often have an issue of boron deficiency in their wheat plants. And it shows as only occurring during seed set. So the image on the left, you can see these are healthy looking plants with no signs of vegetative deficiency. The image next to that, we can see that the minus boron treated, the low boron treated fields have open, poorly set grain head, and yield will be dramatically compromised. That happens for two reasons. Firstly, the pollen grains themselves are not viable. Boron is essential for pollen. Secondly, boron is essential for the formation of the female flower. In this particular case, this deficiency is more common when you have a very high humidity period at flowering, which limits transpiration, limits boron delivery, but you can also induce it with very heavy leaching rainfalls pre-season, which depletes the soil of boron. Corn suffers from boron deficiency in a number of different locations, and here is an example of late season boron deficiency, compromising the ability of the pollen tube to fertilize the ovary, and you have the shriveled grain at the end of the cob itself. The final example I'm going to show you of reproductive effects comes from pea, a legume. Legumes are quite sensitive to boron deficiency. And you can see in the top left image, very poor grain set, pea set. That's an empty pod. You can also see on the bottom right, differing the left panel from the right panel, the impact of boron on nodulation. And here we see that boron deficiency compromises nitrogen fixation, compromises nodule fixation. So in the case of legumes, you have a triple effect acting to cause poor reproductive success. You've got impaired root growth, compromising nutrient uptake, including boron uptake. You have impaired nutrient uptake, compromising the health of the plant, and you have impaired nodulation. And those three factors act together to result in the reproductive failure that we see. Now, I want to finish up. I know I've had a lot of slides in this particular section, but we'll finish up with this one because it also illustrates some important principles. And this is the effect of boron deficiency on fruit and on tubers. Classic old images here from cauliflower, from apples, from mangoes, from peanuts. And one of the very common effects of boron deficiency that unfortunately only tend to show post-harvest, after you've collected these fruits and sent them off for shipping. You get cracking, you get hollow heart, you get a propensity to rotting tissue and deformities that compromise the value. Potato is an interesting example, and it illustrates a principle I've mentioned previously, that you can get impacts on plant growth and development that don't show in the leaves. So this is a potato crop, looks quite healthy, but if you were to pull the potato from the soil and store it for any period of time, you'll see it suffers from very poor potato tuber quality. This is a hidden deficiency, and boron is perhaps the element that you are most likely to get a hidden deficiency. The effects are often only seen post-harvest, after you get those fruits into storage and through transport. It also occurs in apples, it also occurs in avocado, and many of you will have opened up an avocado and seen symptoms such as this. I'm going to summarize, because the point of these particular slides of symptoms that I showed you is to put in your head what is it about these symptoms that can tell us something about the biology of boron. First, deficiency occurs almost exclusively in meristems, whether they're apical, whether they're reproductive, whether they're root. Secondly, you can get leaf deformation. That is a symptom in some species, but much more common is the death of the meristem. Some species show chlorosis, that yellowing of the leaves. This is a consequence of a boron deficiency impacting root growth, causing a deficiency of other elements like nitrogen or phosphorus or iron. Reproductive tissues and fruits are frequently the most sensitive stage. And we'll see later on that's because it's difficult to transport boron to those tissues. Those tissues are growing very rapidly, and hence they have a very large demand. Finally, and a very important concept, is that these deficiencies can occur very quickly, and they can be restricted to very specific organs. And that has implications, as do all of these points, on how we think about managing boron in the field. In this section, what I'm going to talk about is function, uptake, and transport of boron. And we're going to get a little bit more biological and physiological than in the previous ones, but I hope I can do a good job of walking you through the principles. And a lot of what I'll talk about on how plants take nutrients up, how plants take boron up, is relevant for whenever you're thinking about any of the nutrients that are important in agriculture. I'm going to start by giving you the final take-home message, and then we're going to go through and see how these things were derived. Now, as I mentioned previously, the essentiality of boron for plants was established just over 100 years ago in 1923. It took us a long time, all the way until 1996, before we actually determined what boron was doing in plants. And what we've determined is that there are two known and they're closely related functions, but there are a lot of consequences. And many researchers have been led astray by these consequences, thinking of them as primary effects when they were secondary effects. What we know is that boron is a critical structural element in plant cells and in membranes. And I'm going to take you through what that means and a little bit of how we discovered that. So you can go back into the 1940s, and I tell my students this all the time, that there is information of value beyond the age of the internet and lots of clues. So what we're seeing here is some microscope images done in the 80s. And in the top screen are some celery roots that have been grown in the presence of boron. You have two cells that you can see, an upper and a lower one, and between them you see a barrier which represents the cell wall. And that cell wall space is nice and tight and narrow, and you can see those black lines moving through the cell wall, which are plasmodesmata. That is basically communication between neighboring cells. If you look at the bottom slide, which is 72 hours without boron, you can see things have started to fall apart. The cells are very ununiform, the distance between the cells is large, and no longer do you have those plasmodesmata spanning the gap between them, so the cells can't communicate. An analogy for this is that you're laying bricks and you have a very good stable mortar in the top cell, and in the bottom cell you've made a very poor mortar that is not suitable for binding the bricks together, and in this case binding the cells together. You get this disorganization of the space between cells. You get loss of the integrity of the membrane, loss of cell-to-cell communications, and sometimes this can be perceived by the plant as an infection, and then you can get secondary infection responses. So what's going on? It was in 1996 that we finally worked out what was happening here. And so you can think of the cell wall as two layers of fiber, like two sheets of paper, two sheets of fiberglass, two sheets of carbon fiber that are separated by and bound together by a resin, like we do when we're making fiberglass or carbon structures. And what boron does is make sure that bond between the layers is flexible, strong, and suitable for plant growth and development. So boron is the bridge. Boron is what's binding these pieces together in the right conformation so function is correct. Boron also makes that binding between the cell wall and the plasma membrane. And that's what determines the integrity of the cell wall. Now we know the specific chemicals involved, not relevant for this particular talk. But what it does explain to us is why we see these symptoms. If you can't make a stable, properly constructed wall, you will end up with deformations in the leaf tissue like you see. And of course, the most important place where the most cell wall and cell growth is occurring is the meristems. So we can explain what's going on. The deformity in the leaves, the deformity in the meristems and the roots and the fruits is a consequence of this damage to the plant cell wall and the membrane. About how boron moves into the plant, and we'll see later on when we get to specific agronomic management practices why this is important. Now, this is the most complicated piece of my talk today, but I put it in and I thought it was worth keeping because it illustrates some fundamental principles that are important for the uptake of all nutrients by plants. So I'll step you through it slowly. Firstly, we know that boron deficiency can be induced very quickly, and we need to explain that. Secondly, we all know, and those who have worked in the field know, that if we make a mistake when applying boron, we get toxicity. We need to explain why that happens. Thirdly, as I mentioned, boron deficiency affects meristems, particularly reproductive meristems. Why? And finally, boron toxicity symptoms vary by species. All of these 4 points need to be explained. We know, of course, boron deficiency symptoms, which I've shown you previously. One of the things I haven't shown you is boron toxicity, and they're illustrated on the bottom there, and very characteristic symptoms. Now, it was observed almost 50 years ago by a professor in Canada that boron was really quite unique in that it could move between this deficiency stage into the toxicity stage quicker than any other element. And he made the statement at the top of the screen there that boron is unique in the narrow range between deficiency and toxicity. Okay? We need to explain this, but what I'll also do later on is point out how this is only partly true, but it does provide some really valuable lessons on how we should think about managing boron. So the graph on the left, you can see relative yield. On the bottom of that axis, you can see increasing soil applications. Low amounts of boron, you cause boron deficiency. You soon reach adequacy and optimum range, but if you go too far, you get into toxicity. We'll talk about that. Now let's get into some of the physiology and biochemistry, and I'm going to take you through this slowly because it's important and it's a little bit complicated. As you all know, nutrients are taken up by plant roots. Nutrients go into the root through this barrier at the outside of the root, shown here in the purple. If you zoom in a little bit closer, this purple layer, which is the outside of the root system, consists of a membrane, and that membrane has a lot of very important structures in it, shown by these proteins embedded in the membrane. When boron is present in the soil at low to medium levels, so 0.01 to 0.7, which would be relevant for a number of the crops we're talking about, the boron gets through the root, through the membrane and into the inside of the root by passing through these proteins that act as gates and act as regulators. So the boron uptake when boron is at moderate levels in the soil is very tightly controlled. Where boron is unique from all other elements is that if you have high concentrations of boron, it can leak directly through the membrane. And this means the plant no longer has control. To summarize, when boron is at the low concentrations, the plant regulates and controls its movement very closely. When boron is at higher concentrations, the membranes are leaky and the boron can pass freely through. This has consequences, and it helps us explain those effects of the narrow window between the deficiency and the toxicity. Plants have only a moderate ability to adapt to changing boron conditions. Cultivars and species can differ dramatically in how they manage low and high concentrations. These values I show you here are kind of typical for canola. I'm going to go to one level of more sophistication because it's important for how we understand this, but it's also, I think, a beautiful explanation of how the biology of nutrient uptake occurs. So we've zoomed in here to a single cell. And if we look at that purple color the one that's labeled NIP, this is one of the proteins responsible for taking boron into the cell. Now I colored it that purple color because it matches the image on the right, where you also see the purple color. What that means is that those proteins are active in that layer on the outer side. There's a second set of proteins, the green ones here called Boron, that are responsible for the next step of boron transport, taking it from the surface of the root, from the outermost membrane, into the transport system. And you can see in the image on the right how those green colors represent that these particular proteins are on and are active. Personally, I think that's very cool that we can see this level of plant biology at this sort of resolution. If we go to the circumstance of high boron, now we're in the circumstance of boron just leaking in. And now all of these fancy control systems no longer work, and the consequence of that is the toxicity that we see. Now, that's perhaps a little bit more physiological information than you're used to receiving, but you'll see later on why this is really relevant. In the case of canola, which I mentioned before, that the Chinese and also the Australians have been very successful at breeding canola cultivars that are very good at growing in low boron soils. What they have done is manipulate some of these proteins, selected for or bred for particular cultivars that have a much better ability of expressing that purple and that green protein, and hence growing better under low boron conditions. Let's get back to this discussion, okay, where there is this very narrow window between deficiency and toxicity, and explain why it happens. That is that boron is not easily bound in the soil. It mostly exists in the solution phase, and as a consequence, anytime you increase solution concentration of boron, it is available for plant uptake, and it will flood in through these systems. So this is the explanation for why there is this narrow range. But what's important here is that it is all about how much boron is in solution in the soil. So if you make a fertilization mistake, putting 10 times more boron than you think you did, you're putting on a highly soluble, highly permeable element that is going to flush in. As it turns out, plants are not particularly sensitive internally in their physiology to boron, but they are sensitive to this rapidly changing boron availability in the soil. So you make a mistake, put more on than the plant needs, this can happen. In order to understand some of the effects that we saw of boron, that is that it's really the reproductive tissues, the fruits, the roots that are most sensitive, we need to think a little bit about how boron moves around the plant. Now we define a term in plant biology called mobility of elements. And that basically says a mobile element can be taken up by the plant, put into a leaf, and then subsequently moved out of that leaf to wherever in the plant it is needed. An immobile element is a one-way street. So immobile elements can move into tissues, but they can't move out. This is important because if the soil becomes deficient, there is no reserve for the plant to draw upon. You also see differences in what this means for use of fertilizers and the symptoms. If an immobile element accumulates in high concentrations, it will show toxicity in old leaves because that's where it's accumulating. Simultaneously, if boron if a nutrient is deficient, an immobile nutrient is deficient, it will always be the young tissues that show it. Nutrient mobility also affects efficacy of foliar fertilizers. And you think about that, it's kind of clear why that happens. I can get a foliar fertilizer, spray it on a plant. I can satisfy the needs of the tissue that I spray. But if a new tissue grows tomorrow, very little of that applied boron is available for the use by the new tissues. That becomes important for how we manage fertilizers. Very simply, how do you ask the question, is a nutrient mobile or immobile? You can look at the distribution of nutrients in the shoot. If it's fairly uniform from top to bottom, it's generally a mobile nutrient. If it's highly concentrated in the oldest and lowest leaves, that's generally indications of an immobile element. Now, boron is the only element in which different crops, different cultivars behave differently. Okay? In the case of boron, it is an immobile element in many, many species. And for 80 years, all the textbooks said boron is immobile. And it was concluded it was immobile because of those two effects I mentioned. If the meristems become deficient first, that is a sign of immobility. If toxicity occurs in old leaves, that is a sign of immobility. And many crops fit this pattern, but there are crops that don't, particularly tree crops, vegetable crops. Boron is mobile in many of those. Toxicity doesn't occur in old tissues, and you have a uniform distribution of boron throughout the plant. I have a couple of slides here, some nice sort of hybrid images of plants just to illustrate this process here. On the left are species in which boron is immobile or has very low mobility. And remember, that's important because an element that is immobile has the propensity to get deficiencies in young meristematic tissues. So all of the vegetable and tuber crops, with the exception of carrot, there's a little bit of transport that happens in canola, but usually very small. All of the cereal crops, those are immobile. And there's a list on the right of the different species, those that are immobile and those in which boron is mobile. And it changes how we manage boron. We're going to mostly focus on the classic immobile plants because agronomically, those are the most important. So I'm going to summarize this section and what we have learned. We looked at nutrient uptake, we looked at the toxicity patterns, and we looked at flower abortion. And the take-home messages that are important is, firstly, boron uptake is controlled when the supply is low, but it's uncontrolled when the supply of boron is high. Boron deficiency and toxicity therefore respond very closely to soil solution boron. And the most common cause of boron toxicity that I see in agronomy is when somebody makes an application error, putting on 10 pounds when they're meant to put on 1. It's these mistakes that can result in deficiency or toxicity. In most crops, boron is very immobile. And as I said, the consequences of that are twofold. Firstly, it means that deficiencies can occur very rapidly and also most commonly in meristematic tissues. But the second one is an important management issue. When you think about this, it means boron must be constantly available, particularly during those critical growth stages. And later on, we'll look at the growth stages of maize as an example and look at where are the weak points, where are the points where growth is critical, where are the points where a deficiency of boron would have a major impact on productivity. I'm going to wrap it up in this final section and ideally tie together all of the sections. As I said at the beginning, is so that you can understand boron in the context of your particular cropping system. And because of the variability that boron shows with environment, with soils, with crop, with species, it really is up to you to think about these principles and put them back into the context of your particular growing circumstance. I'm going to talk about boron and reproduction. I've mentioned that a number of times, but I want to also highlight here, what is it about the confluence of the biology and the confluence of boron in the environment that results in reproduction being so sensitive? We'll talk a little bit about some of the environmental interactions to pay attention to, and I'm going to use both of those to set the stage for the management principles and the strategies that we must implement. And then I'll summarize up what we've done. So I've shown you all sorts of slides of the impact of boron deficiency on reproduction. But there is that core question of why is reproduction so particularly sensitive to boron deficiency? On the left, we've got images of impacts of boron on the flowering structures. In canola, we see the shattering and the drop of seeds post fertilization. In tobacco, I showed you that example of where the deficiency can result in flower abortion super rapidly. In pistachio, in the top right, I showed you the interaction with cold weather and climate and environment. And in the image there of sunflower, I illustrated how drought and heat stress can cause transient boron deficiencies, in this case reflected in poor seed set. So we'll try and explain these, because I think understanding why and how these occur gives you the information you need to make really smart decisions. A couple of examples. This is some old data from the 80s on red clover, and I put this in to illustrate an important principle. So we have the 6 different rates of boron supply to soil on the left. Then we look at how much shoot dry matter was produced, how many flowers were produced, and then finally what the seed yield is. You'll notice here that you can achieve maximum dry weight growth with even as low, perhaps even lower than 0.25 milligrams per kilogram. But to achieve maximum reproductive growth, either the number of flowers or the seed yield, you needed to go up to 2 milligrams per kilogram. So there's a difference in requirement for reproduction versus vegetative growth. Now in this particular case, you can't actually tell whether it is because reproductive tissues demand more boron or if the fertilization had been depleted by the time you came to flowering. There's a bit of an answer here in this data set that comes from colleagues in Western Australia, in which they derived the boron requirement necessary, the critical value necessary for optimum performance of 3 different organs: the leaf, the pistil, which is the male portion of the flower, and the stamen, the female portion. And 2 varieties, wheat and oilseed rape. And what you see is the leaf tissue is the least sensitive, particularly in wheat. It's indeed very difficult to get a vegetative boron deficiency in a cereal crop. But as you move into the reproductive tissues, the demand for boron is greater. So this is a partial explanation. We also spent some time looking at mobility in the plant. This is another reason why reproduction is sensitive to boron deficiency. Boron mobility is very low. This means that boron in the soil can be taken up by the plant, but it's very difficult for it to be redistributed into the flowers or the fruits as they develop. This means constant supply of boron from the soil becomes supercritical. The other aspect of boron mobility that helps explain why we see deficiencies with particular environmental conditions, in this example drought or heat, humidity or cold, is because both of those factors limit plant uptake of boron from the soil, and since plants can't remobilize what they've previously acquired, this can cause the deficiencies that we see. So when we think about the big question, why is reproduction so sensitive? It's multifold. It's first that the meristems are the most actively growing tissue in the plant. The plant is making new cell walls, the plant is dividing, the plant is expanding. That demands boron be present for cell wall formation. The second is the mobility and the transport. In many species, the movement of boron to apical, floral, and fruiting structures is dependent upon current uptake and water. Water movement drives this. So if you have any circumstance, low transpiration, flow and mobility, competition, you can induce a deficiency. The third is much more agronomic, and that is that on occasions there can be circumstances with lower availability of boron in the soil at flowering. And you can think about this in 2 contexts. An annual crop, you could have a depletion of soil resources as you approach the season's end. Either the fertilizer you put on has been depleted, or the soils have dried and the fertilizer is difficult to take up. In spring flowering crops, you can have a lack of uptake during flowering because the soils are cold, because you've got low activity of the roots. You also have very rapid growth. And if you think about annual crops, you can have dry and depleted soils at the end of the season. So very often, what is happening that causes the high sensitivity of reproductive tissues to boron deficiency is the combination of all three factors. That's important to think about, because in an agronomic context, when you're making decisions, when and how should I fertilize, you have to think about all of these interacting factors. I'm going to summarize the biology a little bit that we've touched upon. We've determined now what the function of boron in the plants. It's critical for plant membranes. Adequate levels of boron are required, but they're particularly sensitive whenever growth is occurring rapidly. Flowering, seed set, seed fill. Roots are also highly susceptible to deficiency, but they're much more difficult to see. In terms of boron nutrient uptake and assimilation, boron has that unique circumstance that it doesn't interact with soil minerals very well. It's uncharged, it's mobile, its uptake is controlled when it's at adequate levels and low levels, but at toxic levels, boron uptake is not controlled. And that's why you have that very narrow window to be between just enough boron and too much boron. We touched on the mobility. Boron in most of our agronomic crops, with the exception of tree crops and some vegetables, is very immobile. And this immobility causes the propensity of plants to become boron deficient if the soil is not supplying boron at very critical stages. The fourth important principle that we outlined is there is an interaction with environment here, and anything that affects plant water use, anything that affects water movement through the plant to reproductive tissues, and anything that affects growth rate has an effect on boron, on its delivery and on its utilization. So, what does this all mean for when we think about how do we frame the management challenges and how do we address boron deficiency and boron management in a logical and a sensible way? I mentioned previously that soil boron tests are really pretty crude and give us only broad guidelines. They can also be complicated by cultivar, environment, and weather conditions. It makes them difficult to use. One of the best guidelines for boron management is plant boron analysis, but more importantly, crop response. What is the history in this region of seeing responses and benefits? That really becomes the best guideline. That, of course, requires an astute observer to know what's going on. One of the principal biological considerations with boron is you have to provide sufficient season-long boron supply. That's really difficult to do if your only application time is at seeding. And if you are relying upon the application of boron at that single time when you plant your crops, you better make sure you have the right product, the right conditions, cooperating weather, and the right release pattern. The ability to put boron on during the season is really very valuable because it allows you to target these times when the deficiency may be present or when the impact of having a deficiency would be supercritical. So fertigation, applying foliars, side dress if you can get out there, all become very valuable management tools. The last one is important to remember. If managed properly, boron toxicity really isn't likely. You get boron toxicity when you make application errors that don't recognize the unique characteristics of boron in the soil and the unique characteristics of boron uptake. Don't get me wrong, management of boron is complex. It's complex because you've got these transient deficiencies, these little windows where it really matters. And they can be very short, a matter of days or even hours in some crops. It matters because different tissues, different stages of growth, and different types of crops have their own specific demand and window of demand. On the top right, I show you an image taken over the top of a canola field where you can see poor performance in some zones and good performance in the other zones. That's a challenge. How do you manage that? How do you measure that? How do you know if it's there? And finally, of course, all the environmental interactions to pay attention to. So it might sound like a particularly daunting task, but as with any agronomic and fertilizer management, it's about thinking about your particular cropping system. So there's an example here of corn and an idealized boron management strategy, or the way you should think about it. Obviously, at germination and emergence, boron is essential for that early seedling growth. It's also true in those soils that if you had a very wet winter and you have a lighter texture soil, you may in fact have very few boron reserves in the soil. The most important phase of boron demand, of course, is flower formation, and making certain boron is available at this time of the year is your most important priority. Of course, there are on occasions things you can't control. A drought just pre-flowering can constrain boron movement. Boron also plays a role in the ultimate grain fill. We saw that in the example of the corn cob. And maintaining a boron supply late can also, in some circumstances, be really important. So if we think about the practical complications and the practical implications, yes, having boron in the soil and in-furrow fertilization is important. But if you have a season-long worth of boron in a single application, you certainly risk the concept of toxicity. And if you have growing conditions during the year that deplete the boron, you also have the propensity to run out of boron later in the season. If you can get on in the middle of the field with side dress or foliars or fertigation, these can be critically important to prevent that potential for a boron deficiency during flowering. And then subsequently, depending on the crop you're in, late season can be important. For tuber crops, this can be a particularly important stage of application. So I'm going to break the final few slides down into some more explicit guidelines. How do you manage vegetative boron deficiency? So this is what might occur if you're growing a vegetable crop, lettuce, etc. Ensuring boron availability at or prior to all key growth phases is essential. The second principle I've mentioned many times is because boron is generally not fixed in the soil, it does become rapidly available to the roots, particularly if it's a soluble boron source. So if you make a mistake in application, you can cause a toxicity. You have to think about that. If you're growing your plants in a very high rainfall, excessive irrigation regime, you can lose the boron through leaching through the soil. That is something to be aware of. Vegetative deficiencies, they can be easily addressed with any high-quality soluble boron fertilizer if the application time is correct. So balancing that availability of boron through the season to match the demand and the growth phases is critical. Similarly, foliar sprays offer quick, targeted correction. Often their effect is short-lived, as I mentioned before. The tissues you spray will be satisfied with the sprayed boron, but new tissues that grow subsequently may not. All of this points to a single theme, and it's one I'll come back to when we talk about managing reproductive boron deficiency. Boron fertilization is highly technical, more so than any other element. You need good advice. You need quality products with reliable, predictable release rates. And you need an agronomist, a soil scientist, an advisor who understands these principles. Let's look at reproductive deficiencies in the last phase. As I mentioned, in many of our crops, this is the key. This is what we need to be sure we are satisfying to avoid the implications that we see there on canola or avocado on maize. The management principles are actually quite similar to vegetative crops. Again, ensuring that you have adequate boron at or prior the reproductive phases is essential. Remember, it's not going to be transported from boron previously taken up and stored in old leaves. The second important point is a tricky one. Now, reproductive deficiencies are difficult to predict because you need to have information on what is still available in the soil, but you also need to have information on when will flowering occur, how rapid will flowering be. And so it makes it difficult to predict, difficult to measure, but we do know reproductive tissues are generally more sensitive than vegetative tissues. And because of all this variability, soil and tissue tests are often not sensitive enough to give us the advice needed to say, do I need to apply or do I not? Third point, deficiencies can occur as a result of limitations in transport of boron to the flower itself, and that is tied in with environmental conditions, but also growth rates. Fourth point, well-timed soil, well-timed foliar applications that are designed to make sure those flowers have adequate boron, which in a tree crop would be last year, but in an annual crop would be this year, are highly effective. And very often, preventative applications to overcome the possibility of a boron deficiency are warranted. That depends a bit on the value of the crop you're dealing with. But again, the same overall take-home message. Boron fertilization is highly technical. You need the advice, and hopefully you need the understanding that comes from this talk to make the decisions that make sense. Quality products, reliable and predictable release rates are the key. I'm going to summarize by going all the way back to the first slide that I showed you and to revisit whether Catherine Warrington in 1923 got it right. The first point, clearly, she got right. Deficiency is always exhibited in the meristem. Apical meristem, reproductive meristem, root meristems. The second one she didn't quite get right, but she was not far off. She stated that boron has a special importance, and probably its function was in the vascular system. Not quite true. The special importance of boron is in the formation of the cell wall and the relationship between the cell wall and membrane function. She didn't know anything about uptake. Her research was well before we could do the fancy genetic analysis that I showed you. The point that I'll add to the summary is that boron uptake is well regulated when soils are low in boron, but it becomes unregulated whenever there's excess boron. Fourth point that she didn't mention is that reproduction has a far greater and unique sensitivity to boron deficiency than almost any other phase of plant growth and development. It's a combination of the biology of flowering and the movement of boron and the rate of growth, but it is also a consequence of agronomy. Will there be boron in the environment during the reproductive phase? Finally, she has the final bullet, which is partially correct. She said a continuous supply of boron is critical. That is certainly a solution, but it's a bit more accurate to say a well-timed and careful supply of boron is critical. And this is where the agronomy, the choice of fertilizers, soil management, soil health, all come to play to make the right decisions. And with that, thank you very much.